Okay, so this is an updated video on mutations for AP Bio, specifically topic 6.7. So when we talk about mutations, there are two major kinds or categories of mutations. There are large-scale chromosomal mutations where whole sections of chromosomes can be deleted, inserted, flipped, put onto different chromosomes. I'm not going to talk about that kind of mutations in this video. Instead, I'm talking about the types of mutations that occur in the DNA sequence that then has impacts or possibly impacts on overall protein produced. And therefore, because proteins determine our phenotypes, we could see if phenotypes change in response. So when we talk about these types of mutations, DNA sequence changes, there can be a couple different reasons why they occur. Now, one could be that there's errors in DNA polymerase. When DNA polymerase 3 is adding complementary nucleotides, it may make a mistake, just like if you have a typo when you're typing up a paper. So incorrect base pairing by DNA polymerase 3 during replication is one source of alterations to a DNA sequence, but it could also be due to external factors such as radiation or reactive chemicals that can cause random mutations. And so when we talk about mutations, though, I think we have this mindset that mutations are negative or harmful. Now, it's important to remember, though, that mutations create changes in proteins and therefore phenotypes. And so mutations are an ultimate source of genetic variation within populations. Now, these mutations can then be acted on by natural selection. If they're a beneficial mutation and confer an advantage, well, then they may increase in a gene pool, versus if they're a harmful mutation, they may decrease within a gene pool. So mutations can be beneficial, detrimental, neutral, and that really comes down to the change in that nucleic acid sequence, and did it have an impact on the protein as well as the phenotype? And was that change in phenotype helpful to the organism, harmful to the organism, or neutral? Right? So when we talk about beneficial mutations, because not all mutations are harmful, some beneficial examples could be a mutation in the PCSK9 gene that actually lowers a person's odds for coronary artery disease, which means they're going to have less buildup of plaque within their arteries, which is then a good mutation to inherit. Another positive mutation would be in the protein that the CCR5 gene that codes for a protein on a white blood cell that the HIV virus uses to enter into the white blood cell. So if that protein is mutated, then these individuals are basically immune to HIV. So those are two examples of beneficial mutations. Now, when we talk about mutations, though, the majority of them really are going to be neutral. If we think about our entire genome, 1% to 2% of our DNA, it only codes for proteins. So 99 to 98% of our DNA is non-coding sequences. So if a mutation happens in a non-coding sequence, then it's not going to have an impact on phenotype, at least directly. And so if it happens maybe in some non-coding genes for microRNA, it might have some other consequences. But generally speaking, if we are talking about having an impact directly on protein folding, then it would have to occur in 1% to 2% of our DNA. And that mutation would also have to occur within an exon to be expressed. So therefore, the majority of the mutations that occur in our cells during replication are going to be neutral. Now, let's go ahead, though, and talk about harmful mutations or detrimental mutations. So a common example would be cystic fibrosis. So there are different mutations that cause different variations of cystic fibrosis, but basically the protein channel where chloride ions travel through doesn't form correctly. And without that functioning chloride ion channel for facilitated diffusion, ions get trapped on one side of the membrane, and then that affects osmosis, creates a hypertonic area. So one side becomes dehydrated, like the water's moving into the cell, and therefore the outside of the cell becomes dehydrated, and a thick mucus builds up, and that causes a lot of respiratory issues. And that's just one example of the outcome of cystic fibrosis. There's more involved in it, but very simply, it would be due to the misfolding of that chloride ion channel. Other examples you could research on your own would be something like Tay-Sachs. Tay-Sachs is a deadly inherited disease due to one point mutation, one switch of a nucleotide, and has detrimental effects on the functioning of lysosomes in the brain and hydrolytic enzymes within those lysosomes. Now, I really love this concept here where it says a mutation is beneficial, detrimental, whether or not a mutation is beneficial, detrimental, or neutral depends on the environmental context. So I'm actually going to use a mutation that I have. Almost 20 years ago, 17 years ago, I had a massive blood clot, a deep vein thrombosis. And I don't want to be dramatic, but there was like a 60% chance a part of it would break off and kill me. So I was young, I was 25 at the time, and so doctors were curious, why did such a young female develop this like very large blood clot? Turns out when they analyzed my DNA, I went to a hematologist and they sequenced my DNA, and I have a mutation for factor V where a protein in my blood clotting pathway, it basically stays active way longer than it should, which then increases my odds of blood clots. Now, if we look at this factor V latent, it's an actual autosomal dominant mutation. So if a person inherits just one copy of it, they're going to be impacted. It will be in their phenotype. And so this mutation is actually due to a single nucleotide switch, a substitution. So when I say the word substitution, you can think about like on soccer, if you have like a midfielder is subbed out for another midfielder, it's going to be in the same position. It's just a switch. So a substitution mutation is switching like an A for a C on accident during replication or due to external factors. And so this mutation, though, caused a misfolding of my factor V protein, and that leaves it on or active for too long. So when I was looking into this, because obviously it affects me, it turns out that this mutation occurred between 20 and 30,000 years ago in a population of humans that was separated from the rest of the world in Europe. So this mutation is actually pretty much non-existent in people of African descent or Asian descent, and it's primarily in people of European descent. So when I look at it more, though, it says anywhere from 12 to 20% of the population may carry this mutation. They won't all get blood clots, but that's a really high frequency for something that could be pretty deadly, right? So when I looked more into it, this mutation in today's society with blood clots and stuff could be pretty harmful, especially when it reacts with different medications. So when you see like TV commercials that are like side effects, blood clots, people with this mutation may be at higher odds for those side effects. But in the past, in prehistoric like human times, this mutation would actually give them a benefit in particular environments because it reduced blood loss during injury, or women who were in childbirth were less likely to hemorrhage and bleed out, or wounds would heal faster. The clotting factors would happen faster, so it'd have less blood loss. It's also hypothesized that it would contribute to less anemia in females. And so this mutation in the past was an advantage, and therefore by natural selection was selected for. But in modern society, if we take prescription drugs or antidepressants or different like birth control pills, etc., those chemicals we are adding into our bodies can have impacts with this mutation and increase a person's chance of clots, which then would make it detrimental in today's society. So mutations and whether they're beneficial, detrimental, or neutral really does depend on the context of the environment. Okay, kind of a long example, but you'll be thankful for that when we get to natural selection. Okay, so let's go ahead though and talk about point mutations, which are those substitution mutations that I was talking about. And they occur when one nucleotide has been substituted for a different nucleotide. So during DNA replication, maybe DNA polymerase 3 is following base pairing rules, but then on accident does incorrect base pairing. So you can see this red C on my screen. That would be a point mutation. So this really only matters, though, if it happens in an exon where it's going to be transcribed and then later translated. So then if RNA polymerase comes and makes a copy of messenger RNA, now we can compare these two RNA sequences. You can see on the bottom strand, which is the original DNA sequence has a U in one particular location, whereas on the top strand it has a G. So the change in codons could have different outcomes. So if we divide this into codons, we start with our start codon, but here you can see the original strand is coding for serine, whereas this new codon with the point mutation codes for alanine. So this example would be actually a missense mutation. Now there are a couple different outcomes that can happen from point mutations. They can be silent, missense, or nonsense. Now if we take the original DNA sequence, and let's say the original DNA was AGC, then there's a mutation where now it's AGA. Well, the RNA would now be UCU for the codon. And if we use the genetic code table that you see here on the bottom left of the screen, if the original amino acid was serine, this new amino acid is also serine. So the mutation is a silent mutation. There was no impact on the polypeptide and on the amino acid sequence. Sure, the DNA was changed, but the amino acid sequence is still the same in that primary structure of the protein, and therefore the protein will fold up normally. Now, in a missense mutation, can I move this? In a missense mutation, this is where you have a change in the DNA sequence that does create a new codon for the mRNA. And when we use the amino acid table, we see that this new amino acid is tryptophan. So original was serine, now it's tryptophan. So the length of the polypeptide didn't change, but you have one switch of an amino acid in the polypeptide or in that primary structure. This is similar to the example I was using for that blood clotting protein. I have one switch of a nucleotide point mutation in exon number 10 that then switched, I think it's like, I forget what it said, arginine to glutamine or something like that. And so now this change though could have an impact or not on that folding of the protein. It really comes down to, does the new amino acid have the same chemical properties as the original? For example, if the original amino acid is polar, is the new replacement amino acid also polar? If that's the case, then it really shouldn't change how that protein folds. However, if the original was polar and the substitution or the point mutation resulted now in a non-polar amino acid, well, that is going to cause different consequences as that tertiary structure forms in that polypeptide. So in missense, it really does come down to the chemical properties of those R groups and whether or not it has an impact on the overall protein folding. But no matter what, a missense mutation does switch an amino acid in the polypeptide. Now, a nonsense mutation, here we have the original DNA sequence was AGC. If that G was subbed out with a T, now the RNA codon is UAG. And UAG actually codes for a stop codon. And so if I look at the three names, they kind of tell us what they do. Silent mutations are going to result in silence. There's no change. A missense, it still has the normal length of the polypeptide. It still folds up. It just may not make sense. May not work the same as before. Maybe it's better in a beneficial mutation. Maybe it's detrimental. Maybe it's neutral. But that protein doesn't fold the same, or possibly doesn't fold the same if the amino acid has a different chemical property. And then there's nonsense mutations, where a nonsense mutation creates an early stop codon. Now, an early stop codon is significant because as the ribosome is reading the messenger RNA codon by codon, it will stop early and that protein won't be finished. And so, if we summarize these three types of mutations, you can see the original amino acid sequence on the left, and in a silent mutation, it's the same amino acid sequence on the right. Whereas a missense mutation, you can see how that proline was switched for a threonine. And so there's a different RNA sequence, and therefore different amino acid sequence. And then in a nonsense mutation, that codon resulted in a stop, or that mutation resulted in a stop codon. It's called a premature stop codon. The ribosome is now going to stop prematurely, and the full length of the polypeptide will not be translated. And so we'll get a shorter protein. So for example, here if we have a mutation that happens, and then we transcribe it into RNA, this right here, UAG, is a stop codon. So then when RNA polymerase begins transcription, it's going to stop right here, and you get a shorter polypeptide that's probably non-functional. Probably not going to work if you're missing the tail end or anything following that stop codon. Probably not going to be a functioning mutation. Now, another type of mutation that exists is called frameshift mutations. And in frameshift mutations, we're going to see a shift in the reading frame. When I say reading frame, I mean how we read codon by codon by codon. So here, if I have the top strand of DNA opened up to do transcription. Okay, awesome. So here's my original amino acid sequence for a polypeptide for this gene. Now on the bottom, if there's some mistakes where some nucleotides get deleted, it could also be inserted. There's two kinds, deletions or insertions. So now if it gets deleted, we can see a shift. Oh man, down here, let me move that. A shift in that reading frame. That wasn't smooth. I'm so sorry. But a shift in that reading frame. Let me try again one more time. There we go. So the reading frame is going to shift over. So now when I divide it codon by codon, let's see if I have to move this. If I divide it codon by codon, I can see that the amino acids start out the same, but wherever that deletion or insertion was, and we shifted all of the nucleotides over in one direction or the next, that now following that insertion or deletions, the reading frame is now different. It's going to be divided into groups of 3 in a different sequence. So now instead of isoleucine, it's threonine. Instead of arginine, it's serine. And so every amino acid after this, it's in a different sequence than the original gene. So here, it just so happened that this frameshift mutation actually created a premature stop codon, a nonsense mutation. And so frameshift mutations are pretty drastic in their consequences, because wherever that shift occurs, anything following that will have a different amino acid sequence, because the codons are now in a different order, basically. Okay. So if we look at frameshift mutations, we could have the wild type is what is like the original sequence. And we can see here in a base pair deletion, if you delete just one and the rest of the codons move over, now when you group them into 3s, you have a completely different sequence of codons. In a base pair insertion down in the bottom right, if you add an extra base, then that's going to shift everything in the other direction, but you're still inserting a base, and the reading frame will be shifted. Now, one type of frameshift mutation that maybe isn't so bad is when you either insert or delete codons in groups of 3, or not codons, nucleotides in groups of 3. You can see in this bottom screen, if you remove 3 whole nucleotides, you're basically just removing a codon, and therefore the polypeptide will be 1 amino acid shorter. Or if you insert 3 nucleotides, the polypeptide will be 1 amino acid longer. But the codons following that will also be in the same sequence because you removed a group of 3. Now, I guess it depends, though, on which amino acid was removed or added, how that will impact the overall shape. So let's go ahead then and look at this words for anyone. Frameshift mutations occur when 1 or more nucleotides are inserted or deleted, causing the reading frame to be shifted. Okay, so I think my last concept is what happens when there is a frameshift mutation in an intron region of a gene. So here I've highlighted the introns in red. And so if I copy the RNA and then I remove the introns during RNA processing, here's my mature mRNA sequence. Awesome. Now here, if there is an insertion or a deletion, a frameshift mutation in an intron, so let's say I delete that, or a couple of them, and so sorry, that was a little off, but if I deleted a few introns from, if I deleted a few nucleotides from an intron, and now the reading frame was shifted, the question becomes, does it matter if it occurred in an intron? Because mutations happen in the DNA. Mutations aren't happening in the RNA. So this is in the DNA before pre-mRNA is even made. So once we make that pre-mRNA, you might see this and be like, uh-oh, that's a stop codon. However, we're going to go through RNA processing, and mature mRNA is going to RNA processing is going to remove those introns to produce mature mRNA, and then the RNA sequence is the same for both of these. So mutations that happen in introns, whether they're frameshift or substitution, aren't going to have any impact on the actual protein simply because the introns are removed before transcription and translation occur. Okay, so I think that's the end of my PowerPoint. Yep. Hopefully this was helpful for you, and great job.